Hello and welcome again to our short biochem course. And you're used to the fact that we're doing a lot of nice things during these videos, and we're studying a lot of nice things in biochemistry. And this time I have decided to tell you a little bit about carbohydrates. So I have chosen one very sweet topic for today. So speaking of carbohydrates, those are compounds widespread in plants, and they are actually synthesized as basic products of the process of photosynthesis. There are some other names which are used for carbohydrates, like saccharides or sugars. I think that is the most popular common name, or glycides, which is a kind of old name for carbohydrates. According to the chemical structure, carbohydrates can be defined as polyhydroxylaldehydes or polyhydroxyl ketones, or complex molecules forming polyhydroxylaldehydes or polyhydroxyl ketones by hydrolysis. They are very important for linkage of the energy exchange cycle in nature. So in plants, carbohydrates are synthesized from these simple molecules like water or carbon dioxide, and the energy for the transformation of those molecules is obtained from the sun energy, and those inorganic substances are actually converted or organified to organic substances, which are nothing else but kind of unique hydrates of carbons, meaning that to the carbon atom a molecule of water is attached. And that is the reason why we call this group of molecules carbohydrates. So water plus carbon dioxide plus energy forms the hydrates of carbon atom and oxygen. Regarding the carbohydrates which are synthesized by photosynthesis in plants, we have to mention cellulose, which is the major structural polysaccharide in plants, and starch, which is the storage polysaccharide in plants. Human organism is supplied with carbohydrates by diet, but if for any reason our cells are lacking carbohydrates or lacking glucose, which is the major representative of sugars in our body, our cells actually have the ability and mechanisms to synthesize glucose and its metabolites from simple non-carbohydrate substances in the metabolic pathway called gluconeogenesis, which we are going to discuss in details in some other videos. So, carbohydrates are really important energy source, and we say that they are kind of short-term energy sources because you get, the quick energy, not long-lasting energy, and you get it from glucose or glycogen, which is a unique polymer of glucose molecules. And this energy can be used for the maintenance of the basic metabolism of the cell. Also, carbohydrates have a structural role as well. So they are components of nucleic acids, Pantosis, ribose, deoxyribose. They are found in the structure of glycoproteins, glycolipids, and some other molecules. They have a role in intracellular communication because carbohydrates are important components of receptors, which are actually glycoproteins, and so on and so on. So to summarize this introduction to carbohydrates, we can say that carbs actually contain carbon, hydrogen, and oxygen in a precisely defined ratio 1 to 2 to 1. 1 carbon, 2 hydrogens, and 1 oxygen. Actually, they are described as molecules in which carbon atom in sugar molecule is surrounded by the molecule of water, and the simple sugar molecule can be represented by the formula which you can see from the slide, CH2O n times. So for example, the molecule of glucose, which has 6 carbon atoms, it's going to be C6H12O6, which is going to be the same for fructose as well, and some other isomers. So according to the complexity of the molecule, carbs are classified in three major groups. The first are simple sugars called monosaccharides, for example, glucose, galactose, mannose, fructose, and so on. They cannot be hydrolyzed to produce more simple compounds. Then disaccharides and oligosaccharides, they are polymers of 2 to 10 monosaccharide units. For example, maltose, lactose, saccharose, which are disaccharides, dextrins, oligosaccharides, and so on. And finally, polysaccharides, which are polymers containing large number of monosaccharides in the structure. For example, starch, cellulose, glycogen, pectin, and so on. Monosaccharides are simple sugars, so they cannot be hydrolyzed, to more simple carbs. And they are actually the building bricks of complex carbohydrates. They are crystal compounds, they are soluble in water, and most often of a sweet taste. They can be classified in two ways. According to the functional group, which is found in the structure, they can be classified to aldoses containing an aldehyde group, so they are marked as polyhydroxyl aldehydes. And ketoses, because they contain keto group, so they are marked as polyhydroxyl ketones. The presence of free aldehyde and free keto group actually provides reducing properties to carbohydrates. The other classification is according to the number of carbon atoms. So after the prefix aldo and keto, you can say trioses if they contain 3 carbon atoms, tetroses containing 4 carbon atoms, Pantosis 5 carbon atoms, hexoses 6 carbon atoms, heptoses 7, octoses 8, nonoses 9, and so on. Actually, from the point of our metabolism, we have trioses, tetroses, pentoses, hexoses, and those marked with red are actually the most abundant ones in our organism, but definitely the most important are those with 5 and 6 carbon atoms in the molecule. Monosaccharides with the same number of carbon atom, they actually exhibit, they have the same molecular formula, but they have this different structure. And from your organic chemistry course, you know that those molecules are marked as structural isomers. So actually, when talking about carbs, we're gonna talk about 4 different types of isomerism, and definitely the first one is structural isomerism. For example, glucose and fructose are different molecules. One is aldose, the other is ketose, but they have different rearrangement of OH group, but they contain 6 carbon atoms, and the molecular formula is C6H12 O6, so they have the same formula, but those are different molecules with different structure, so they are considered to be structural isomers. Let's do some examples. So trioses, they are oxidative products of 3-hydroxyl alcohol glycerol. So we have 2 representatives. We have glyceraldehyde and dihydroxyacetone. The first one is the sweet colorless crystal substance. The other one is a white crystal powder of a sweet, cooling taste and specific smell. And both are intermediates in the carbohydrate metabolism or glucose degradation. And here you can see the structure of those. So this is the structure of glyceraldehyde, and this is the structure of dihydroxyacetone. So there are some things that you can notice from the structure of those two. If you take a look at the structure of glyceraldehyde, you notice that there is one asymmetrical or chiral center, and this is the carbon atom to which 4 different groups are attached. So when a molecule has at least one chiral center or chiral carbon atom, it's considered to be optically active compound. And when a compound is optically active, it can be represented by 2 isomers, which are marked as optical isomers or enantiomers, and they are marked as D or L isomers. So if you take a look here, the OH group on the right side of the glyceraldehyde molecule is in the D form of D-glyceraldehyde, and if OH group is positioned on the left, then we say, okay, it's L form and this is L-glyceraldehyde. If you take a look at the structure of dihydroxyacetone, you can notice that there is no asymmetrical carbon atoms in the structure of this molecule, so this is the only monosaccharide which is not optically active. Speaking about the presence of D and L forms of sugar in human organism, we can conclude that D optical isomers of monosaccharides are the only important ones. L monosaccharides are not going to be found in our organism. Moving on to tetroses, D-erythrose is worth mentioning, containing an aldehyde group, formed in our body in the metabolic pathway called hexose monophosphate pathway in the form of erythrose 4-phosphate. Moving on to Pantosis, we have two famous representatives, which I think everybody knows, D-ribose and D-deoxyribose, and we have some others like D-arabinose, D-xylose, and so on. D-ribose, we all know, it's found in RNA, it's found in certain coenzymes, which I have already mentioned, NAD, NADP, FAD, FMN, and 2-deoxy-D-ribose is found in DNA. If we take a look at the structure of some of those Pantoses, we can compare structures of D-ribose and D-arabinose. In D-ribose, each chiral OH group is on the right side, and in D-arabinose you have one OH group at C2 on the left side, and the rest of the structure is definitely identical to the one in the molecule of D-ribose. So when you have two molecules which actually differ only by the position of one OH group in a molecule, then they are marked as epimers. So this is the third isomerism that we are mentioning in carbs. So it was structural isomerism, it was optical isomerism, enantiomers, D and L forms, and now we have epimers. So arabinose is then considered as 2-epimer of the ribose. The next important thing is that in an aqueous solution, these monosaccharides are not found in their acyclic forms, but rather they tend to make cyclic structures. They tend to make heterocyclic rings in a way that an aldehyde group from a molecule reacts with OH group from the farthest chiral carbon atom, thus forming heterocyclic ring with oxygen in the structure. In this case, we're going to have 5-membered heterocyclic ring with oxygen called furanose. So this is going to be D-ribofuranose. Those formulas are either Fischer's or Hayward's cyclic structures and formulas. And those Hayward's formulas are actually the common way, an accurate way to represent monosaccharides in aqueous solutions. There is a rule when writing those Hayward cyclic structures. The OH group, which was on the right side, it goes below the ring, and the groups which are on the left side, they go above the ring. So in the case of D-ribose, OH group is on the right on C2, so it goes down. H is on the left, so it goes up. On C3, OH is on the right, so it goes down. H is on the left, it goes up. In C4, you have just H and the 5th carbon atom. And here is the oxygen atom. But take a look here. One of the results of this reaction is not only the formation of heterocyclic ring, but rather one of the results of this cyclization is the presence of hemiacetal or semiacetal group on carbon number 1. And this OH group can be positioned either below or above the ring. If it's below the ring, it's marked as alpha. If it's above the ring, it's marked as beta. So in short, those alpha and beta forms are marked as anomers. So we have alpha D ribofuranose or beta D ribofuranose depending on the position of hemi or semi-acetal OH group at C1 carbon atom. If we move on to hexoses, the most important and abundant monosaccharides of all, and aldohexoses which are worth mentioning are D-glucose, D-galactose, D-mannose, and one ketohexose, which is D-fructose. And we can summarize our isomerism in this figure. So here is the molecule of glucose. In order to understand its structure properly, it's valuable writing in a cyclic form, but we all know that the accurate way to represent it is the cyclic form written below. So we have 6 carbon atoms, aldehyde group, and OH groups, right, left, right, right. When you're writing the optical isomer, when you want to write L isomer, you have to change the position of all groups on chiral carbon atoms. So if you take a look, this is chiral carbon atom C2, so it was OH was on the right, now it's on the left. C3 is also chiral OH group on the left, it was now it's on the right. C4 chiral carbon atom OH group on the right, now it's on the left. C5 carbon atom also chiral, the last one, OH was on the right, now it's on the left in L-glucose. And if you ask me, if there are more chiral centers in the molecule than one, how can I predict which of the isomers are going to be D or L? So we're following and we're analyzing the position of the OH group on the chiral carbon atom, which is the farthest from the functional group. So from an aldehyde group, this C5 is the farthest, so by the position of OH group at C5 we can conclude about the optical isomerism. So this one is going to be D-glucose and the other one is going to be on the left side. Then the epimers. So if I have a molecule of glucose and if I change the position of OH group on only one carbon atom, so here it was on the right, now it's on the left, I have 2 epimer of D-glucose and this monosaccharide is marked as mannose. And one more example, if the OH group is positioned oppositely on C4 only, we have 4 epimer of D-glucose and that is nothing else but the molecule of galactose. And of course, if we are writing the cyclic forms, OH group on C2 is right, so it goes down below the ring. OH group at C3 is on the left, so it goes above the ring. OH group at C4 is right, so it goes below, and so on. So this is the molecule of D-glucopyranose, because you have the formation of 6-membered ring with the oxygen in the heterocyclic ring, which is similar to pyran, so it's pyranose. And again, we have alpha and beta options, so I have written both. If the C1 OH group is below the ring, then it's alpha, and if C1 OH group is above the ring, then it's beta D-glucopyranose. And let's say a few general information about these. So glucose, widespread, in nature, in humans, it's also marked as glucose, dextrose or grape sugar. It's found in fruits in free form, it's found in human body in free form, and the reference values ranges from approximately 3.3 to 5.5 millimoles per liter. It's the component of different disaccharides, oligosaccharides, polysaccharides. It's of sweet taste, it's very soluble in water, like 180 grams of glucose per 100 grams of water. It's optically active molecule with a specific angle of rotation of 52.5, and we're going to use it for our practical exercises later on. So galactose found in disaccharide lactose, which is a dairy sugar. It's found in complex lipids like cerebrosides, sulfatides, mucopolysaccharides, keratin sulfate, chondroitin sulfate, proteoglycans, and glycoproteins. And it's obviously 4-epimer of D-glucose. And mannose is also kind of spread in certain glycoproteins, in sialinic or neuramic acid, and in gangliosides. Fructose is a ketohexose. If you take a look, it contains keto group, not an aldehyde group, so it's a structural isomer of glucose. It's also called fruit sugar, so it's found free in fruits and honey, and it's the component of the disaccharide saccharose. So when making a cyclic structure, you have the reaction between the keto group and the last chiral OH group. So you're going to get a 5-membered heterocyclic ring with oxygen, so it's got a furanose form. And again, the same rule, C3 left goes up, C4 right goes below the ring, and so on. Oligosaccharides are kind of more complex saccharides, polymers of monosaccharides containing 2 to 10 monosaccharide units. And they can be classified to disaccharides, trisaccharides, tetrasaccharides, pentasaccharides, and so on. The common characteristic is that monosaccharide units are attached to each other by glycosidic bond. Disaccharides are the most abundant oligosaccharides, and important for us to mention them right now. So they are solid crystal substances. They are easily soluble in water. And monosaccharide units are attached by the formation of O-glycosidic bonds. And generally speaking, there are 2 types of O-glycosidic bonds in disaccharides. The first one, and more abundant one, is maltose type of bonding. Disaccharides containing this type of bonding, they exhibit reducing properties like maltose, lactose, and so on. And there is another type, which is marked as trachalose type of bonding, which does not exhibit reducing properties, and we will find and see this type of bonding in a molecule of saccharose. So let's start from the beginning. So maltose, or alpha-D-glucopyranosyl, alpha-1,4, alpha or beta-D-glucopyranose is the chemical name. So it consists of 2 molecules of glucose, and those are connected by so-called alpha-1,4 glycosidic bond. Let me explain the formation of the glycosidic bond using this example. Alpha means that one molecule of glucose will give the semiacetal OH group at C1, and this one is in alpha position, so below the ring. So this is alpha 1 OH group, and OH group at C4 of another molecule of glucose. So this is the second molecule of glucose, and its OH group goes into the reaction. The molecule of water is released, and this unique oxygen bridge is nothing else but the glycosidic O-glycosidic bond, and due to the components which were involved in the formation of this bond, we can say it's alpha-1,4 glycosidic bond. Notice that here only one semiacetal group was involved in the formation of the bond, but the semiacetal group at C1 of the second glucose molecule is still free, and due to the fact that this one is carrier of reducing properties, the whole molecule exhibits reducing properties. Then lactose, or dairy or milk sugar, it's beta-D-galactopyranosyl, beta-1,4 alpha or beta-D-glucopyranose. So it consists of molecule of galactose and a molecule of glucose attached by beta-1,4 glycosidic bond. So here OH at C1 was beta above the ring, and OH at C4 was below, is always in glucose below the ring, so it's beta-1,4 glycosidic bond. Those 2 sugars, maltose and lactose, are so-called reducing sugar because here in lactose as well we have free semiacetal group. But Speaking about the sucrose molecule, or white table sugar or cane sugar, it consists of molecule of glucose and molecule of fructose, but they are connected by the trachalose bond, or called alpha-1 beta-2 glycosidic bonds. And this bond is formed, if you see, upon the reaction of both semi-acetyl groups at alpha, which was alpha on C1 of glucose and beta on C2 of fructose. So we don't have another OH group, reactive oxygen group here, so sucrose does not exhibit reducing properties. Speaking about polysaccharides, they contain large number of monosaccharides, which are connected by those O-glycosidic bonds of different subtypes. But this time they are insoluble in water, especially in cold water, slightly more soluble in warm water, and they do not have sweet taste this time. They exhibit no reducing properties because they actually lack those free semi-acetyl groups. And you will see later on that, for example, in a molecule of starch or glycogen, you're going to have just one semi-acetyl group and you're going to have thousands of glucose molecules, which is definitely not enough for the molecule to exhibit reducing properties. So polysaccharides are classified in two groups, homopolysaccharides. Actually, they contain only one type of monosaccharides in the structure, like starch, cellulose, glycogen, agar, pectin, and so on. And we have the group of heteropolysaccharides, which are mucopolysaccharides and sugar components of proteoglycans, and they contain two or more monosaccharide types in those basic units, predominantly disaccharides, which can further be polymerized as disaccharide plus disaccharide plus disaccharide, but they contain different types of monosaccharides in the structure. They might exhibit storage function, structural function, they can have mixed character, that depends on their structure and properties. One of the most famous polysaccharides is amylose, is starch or amylum. It's the storage polysaccharide in plants, and it's similar to the glycogen in animals. It contains two domains or two parts. The first one is amylose. I'm sorry for my typing mistake. M-E-L-O-S-E. So I have swallowed one L. So amylose is a less represented component, and if you can see here from the figure, it's linear part of the molecule. It contains glucose units connected by alpha-1 4 glycosidic bonds. And this linear part of the molecule amylose, it tends to form, let's say, the secondary structure, thus forming the helix. And in those coils of the helix, iodine molecules can be incorporated to form an indigo blue color, which is a reaction used for proving the presence of starch in the mixture. Then we have amylopectin, that is the second predominant part of starch. Approximately 75% of the total mass of the starch is amylopectin, so it has like several thousands of D-glucoses. But in amylopectin you have two domains, because that is the branched part of the molecule. So you have glucoses attached to each other by alpha-1 4 glycosidic bonds, because they're forming some linear fragments. And then at the branching points we have one more type of bonding, which is alpha-1 6 glycosidic bond, which is formed like here, alpha OH group at C1 and OH group at C6, so they tend to form alpha-1 6 glycosidic bond. Glycogen is the major storage polysaccharides in animals and humans, and the largest amount of glycogen are found in liver and muscles for maintenance of the homeostasis of glucose or for muscle contraction. It's a branched molecule, even more branched than starch, and structure is similar to amylopectin. Finally, to mention cellulose, which is a large linear polysaccharides containing several hundred, several thousands of glucose molecules attached by beta-1,4 glycosidic bonds. In humans, cellulose cannot be digested because of the lack of the specific enzymes which can hydrolyze this beta-1,4 bond. Moving on to heteropolysaccharides, we can say that apart from the sugar moiety, they can have, and they have, small quantity of proteins found in the structure. So the common characteristic of those molecules are that they can bind large amounts of water. So they tend to form gels, which are the basis of all supportive tissues like connective tissues, collagen fibers, and so on. So they are viscous, and they are the basis for the mucous membrane integrity. So and they kind of line body passages with a very important protective role, and thus they are also marked as mucopolysaccharides. I have provided you with a couple of examples which are important to be mentioned and explained. Hyaluronic acid, everyone talks about hyaluronic acid nowadays for the maintenance of the proper strength and elasticity of our skin and supportive tissues. So it's the largest glycosaminoglycan, and it's a polysaccharide containing two types of monosaccharide units, which is D-glucuronic acid and acetyl D-glucosamine. So several hundreds or several thousands of those disaccharide units are found in the molecule of hyaluronic acid. And this one is the only glycosaminoglycan which does not contain sulfates in the structure. It's soluble in water, it's really, really viscous, and it makes the basis for all connective tissues, for synovial fluid, for intraocular fluid, umbilical cord, and so on. Next one to be mentioned is chondroitin 4 and 6 sulfate. They are the most widespread. They contain, again, D-glucuronic acid, but this time an acetyl D-galactosamine and plus sulfatated like 4 or 6 sulfate. And they are found in the structure of cartilage, tendons, ligaments, and so on. The next one, keratin sulfate and acetyl glucosamine and D-galactose 6 sulfate are found in the structure, and it's found in cartilage and cornea. Dermatan sulfate, of course, its name says it's found in derma or skin. So it contains an acetyl D-galactosamine sulfate and L-iduronic acid. And apart from skin, it's also found in blood vessels, heart valves, and so on. And finally, one interesting molecule, heparin, which is used as an inhibitor of coagulation. It's also found in mast cells of estuaries, liver, lungs, and skin. The content is not described to the fullest, so it is suggested that it's formed from sulfates of D-iduronic acid and N-sulfo-D-glucosamine 6 sulfate. So anyway, that was the short or less short, or short or long, whichever you want, story on carbohydrates. I hope it was not that complicated. We have reminded ourselves about numerous things from organic chemistry, a lot of isomerism, a lot of other properties, a lot of representatives, different nice structures of molecules, and we have made a nice introduction to be able to understand metabolic processes of carbohydrates in the way that they should be understood. So until some other stories in biochemistry, have a nice rest of the day. Stay safe, stay healthy, and bye bye.